Imagine trying to understand a library with millions of books, but no organization system. That's the challenge scientists face before taxonomy. Taxonomy is the science of naming, defining, and classifying groups of biological organisms on the basis of shared characteristics. It's like creating a giant library catalog for all living things, sorting them into groups based on their relationships. This system helps us make sense of the vast diversity of life on Earth. For example, instead of just saying cat, we can be more specific and say Felis catus, which distinguishes it from other felines like lions, Panthera leo, or tigers, Panthera tigris. Taxonomy allows us to see the connections between different species and understand how they evolved. Without taxonomy, studying biology would be incredibly difficult. It'd be like trying to understand a complex machine without knowing the names or functions of its parts. Taxonomy provides the framework for organizing and studying life in all its forms. Inside almost every cell of your body lies a complex molecule called DNA, short for deoxyribonucleic acid. This molecule is the instruction manual for building and maintaining an organism. Imagine DNA as a long, twisted ladder. The rungs of this ladder are made up of pairs of chemicals called bases, and the order of these bases is what makes each person's DNA unique. This sequence of bases is like a code that determines your traits, from the color of your eyes to your risk of certain diseases. DNA is passed down from parents to offspring, which is why children resemble their parents, but are not identical copies. The discovery of DNA's structure in 1953 revolutionized biology. It opened up a whole new world of understanding about how life works at the molecular level. Everything around us is made up of elements like carbon, oxygen, and nitrogen. These elements are the building blocks of matter. But did you know that elements can exist in different forms called isotopes? Imagine having a basket of apples. All the apples are essentially the same fruit, but some might be slightly bigger, smaller, or have a different shade of red. Isotopes are like that. They have the same number of protons, which defines the element, but a different number of neutrons, giving them slightly different properties. Some isotopes are stable, meaning they remain unchanged over time. Others are radioactive, meaning they decay and emit radiation. These differences in isotopes might seem small, but they have a big impact on how atoms behave in chemical reactions and biological processes. Now, how do these two seemingly different worlds of DNA and isotopes connect? Isotopes are incorporated into DNA and other biological molecules through various metabolic processes. For example, carbon isotopes from the food we eat become part of our DNA. By studying the ratios of different isotopes in biological samples, scientists can learn a great deal about an organism's diet, migration patterns, and even past environments. This is like using isotopes as a fingerprint to trace the history of an organism or a biological process. The study of isotopes in DNA is a powerful tool for understanding life on Earth. It helps us answer questions about evolution, ecology, and even the origins of life itself. This field highlights the interconnectedness of different scientific disciplines, as it draws on knowledge from biology, chemistry, and physics. As technology advances, we can expect even more exciting discoveries in this fascinating area of research.